You probably remember Gregor Mendel and his pea plants from biology class, right? The simple, neat rules of dominant and recessive genes. But what happens when life, in all its amazing complexity, decides to just break those rules?
Well, it turns out most traits don't actually follow that simple Mendelian playbook. Genetics can get way more complicated. And honestly, the exceptions? That's where things get really fascinating. They're not mistakes. They're more like road signs pointing to a much, much deeper biological story.
Okay, so this one experiment, it really captures the whole mystery. Scientists crossed these two types of fruit flies, expecting one thing based on Mendel's laws, but they saw something completely different. And in science, when your predictions are this far off, it's actually a super exciting sign that you're about to discover a brand new rule.
So, before we can really get into the rule breakers, we've got to go back and remember the rules themselves. And it all starts with Gregor Mendel.
He gave us this super clean, predictable world of inheritance. In his world, different versions of a gene, we call them alleles, are either dominant or recessive. So if you cross two purple flowers that both carry a hidden white allele, you get a totally predictable 3-to-1 ratio of purple to white babies. It was a simple, elegant model that became our foundation. But what happens when the relationship between those alleles isn't so black and white, or, purple and white? Let's dive into our first set of exceptions and see how this all starts to get a little more complicated. All right, first up, we have something called incomplete dominance. This is what happens when one allele just can't completely overpower the other one. Instead of a clear winner, they kind of meet in the middle. The easiest way to picture this is to think of it like mixing paint. You cross a red flower with a white flower and boom, you get pink offspring. That's incomplete dominance in action. The physical trait you see, the phenotype, is literally a blend of the two parents. And then there's its cousin, co-dominance, which is a bit different. This isn't a blend. This is both alleles showing up to the party and being fully expressed, right next to each other. They're not blending, they're co-stars, sharing the spotlight. The absolute perfect example of this is human blood type. The allele for type A blood and the allele for type B are co-dominant. So if you get one of each from your parents, you don't get some kind of blended blood type. Nope, you get type AB blood, where both the A and the B markers are present on your red blood cells. Both traits fully visible at the same time.
Okay, so we've seen that the relationship between alleles can get complex, but there's another whole other layer to this. Turns out a gene's physical address, its actual location on a chromosome, can completely rewrite the rules of inheritance.
And that brings us to sex-linked traits. These are genes that are physically located on our sex chromosomes, the X or the Y. And because biological males who are XY and females who are XX have different combos of these chromosomes, these traits just don't follow those typical Mendelian patterns.
Now, a family tree like this, a pedigree, it's like a geneticist's treasure map. Just look at this thing. Notice how the trait almost always pops up in the males across the generations? That's because males only have one X chromosome. So if they get just one copy of a recessive X-linked allele, they'll show the trait. Females, on the other hand, have two X's, so they need two copies, which makes it way more likely for them to be carriers without showing the trait themselves.
And this right here gets to the absolute heart of the biggest challenge to Mendel's entire framework. See, his principle of independent assortment basically says that the gene for, say, seed color gets inherited totally separately from the gene for plant height. But what if that isn't always true?
And this is where we meet Thomas Hunt Morgan and his famous fruit flies. He did this cross. He took a female fly that had different alleles for two traits, body color and wing shape, and crossed her with a male that was recessive for both of those traits. The result of this seemingly simple experiment would go on to change genetics forever.
So, if Mendel's rules were the whole story, if his law of independent assortment held true, Morgan should have seen 4 different types of offspring in basically equal numbers, a nice clean 1-to-1-to-1-to-1 ratio. That was the prediction.
But that is not what he saw. I mean, not even close. The numbers were totally skewed. The flies that looked just like their parents were way, way overrepresented, and the new combinations, the ones we call recombinant types, were super rare. Something was clearly going on here, some invisible force keeping these genes from sorting themselves out independently. Yeah, let's just put a number on this. Only about 17%, that's it. Only 17% of the baby flies showed a new mix of traits.
If the genes were truly independent, that number should have been 50%. A gap that huge is a massive red flag in genetics. It's basically screaming that these two genes are somehow tied together. So what was going on? The answer, the big reveal, is something called gene linkage. It turns out the genes for body color and wing shape are physically located on the same chromosome. Think of them like two people sitting in the same car. When the time comes to make a new generation of cells, they tend to travel together. They're way more likely to be inherited as one single package deal.
And you'd think that's complicated enough, right? Well, the plot thickens even more when we realize that not all of our DNA is even located in the nucleus. Let's take a look at some of the other hidden rules of the genetic code.
Get this, our mitochondria, the little powerhouses inside our cells, have their very own separate DNA. And this mitochondrial DNA has a really unique rule of inheritance. It is passed down exclusively from the mother, because the egg cell is what provides all the organelles for the developing embryo.
Okay, now look at this pedigree. See how different this pattern is from the sex-linked one we saw earlier? Here, if a mother has the trait, all of her children get it, every single one. But if a father has it, none of his children do. That right there is the unmistakable fingerprint of mitochondrial inheritance.
And for our last little curveball, what if one single gene could do a bunch of totally unrelated jobs? Well, it can. That's called pleiotropy. For example, the one single gene mutation that causes sickle cell anemia doesn't just change the shape of red blood cells. It also, amazingly, provides resistance to malaria and can affect everything from your spleen to your bones. It just shatters that simple one gene, one trait idea we all learned first.
So, okay, we've gone through all these exceptions, these so-called rule breakers. And you might be thinking, wow, that just makes genetics messy. But here's the thing about science. Anomalies are actually opportunities. And these clues, they led to one of the biggest breakthroughs of all time. This, right here, is the ultimate payoff. Thomas Hunt Morgan figured out that the frequency of recombination, that 17% number we saw with the fruit flies, was directly connected to the physical distance between genes on a chromosome. By studying how often linked genes got separated, he and his team could literally create the very first maps of chromosomes, showing the exact location of genes. The rule breakers weren't breaking genetics at all, they were revealing its geography. From simple pea plants to mapping the very code of life, the story of genetics has always been about embracing the exceptions. And it really makes you wonder, doesn't it? What fundamental rules are we following today that future discoveries might just reveal as another beautiful exception to an even deeper law?